All right, everyone. Thank you so much. Welcome back. Thanks again for staying with us today at MIDDD Plus 2023. In this session, Zack Kenz, one of our senior scientists from our ILDsym Services division. It's been 3 in a row with Liesel, Jeff, and now Zack. Will present an overview of a recent ILDsym poster. As a reminder, please do ask questions in the Q&A tab. For technical issues, use the chat feature. Before I pass this off to the speaker, reminder that there is a poll question we would love you to participate in. Thank you, everyone. Thank you, Zack. Please take it away.
All right, great. Yeah, thanks, Andy. So yeah, if you've seen Liesel and Jeff's talks earlier, they set this up very well in terms of some of the ideas behind our QSP, QST modeling. And the sort of application we're going to look at, which is ILDsym, so interstitial lung disease simulator in the context of systemic sclerosis, interstitial lung disease, so SSc-ILD. And so we're going to look at the modeling and development process behind the platform and sort of how we've built the platform, understood the pathophysiology, and ultimately represented patients that can be used to assess potential drugs or drugs that are in the development pipeline. So we'll go through, a sort of broad overview of that process, which is what the poster that we presented at PharmSci last year was designed to address. So present kind of that broad overview, methods, and then our validation in terms of patients, and then also some simulated treatments in terms of standards of care. So we'll don't try to take this all in. We're going to go through each of these stepwise and address each of these pieces. So to get into the development, when we look at the modeling in terms of a disease area, SSc-ILD in particular, we start with understanding the populations that may be impacted. And the pathophysiology. So in this case, SSc-ILD, we're looking at individuals that are presenting later stage in life. So here, around 55. In terms of gender distribution, we're looking at proportionately higher in women, and a fibrotic lung disease that has high mortality rates, which is why there's interest in trying to help those patients that have SSc-ILD. And it's an inflammatory and fibrotic presentation. So there's components of each of those in the pathophysiology. And then ultimately, those patients have respiratory function declines that can lead to, if the disease progresses, to decreased function overall. So we can see that in terms of survivability rates. That figure is a little bit hidden, but patients with patients who have lung-related issues have lower survival rates, and you can see it in terms of changes in the lung physiology. There's variable course in terms of patients' progression. So some patients who have lower fibrosis, greater fibrosis, that can change the course of the disease. And we want to capture all these different aspects in terms of the actual model development. And I'll dig into this diagram a bit more, but we want to be able to predict treatments, effects of treatments on fibrosis, inflammation, the lung alveoli, and then the ultimately relevant clinical outputs. So that's what we're trying to get at and discuss. And the software that's been developed is intended to support that treatment assessment. And so novel treatments can be added in and can support hypothesis development, particular mechanisms of action, all the sorts of things you'd be looking at in terms of potential drug development. So let's start digging into the pathophysiology itself. So in terms of the things going on in the lung, we're looking at, say, areas of normal lung. So patients do have capacity within their lung. That's what's responsible for the residual respiratory function. And then patients have areas of what are called ground glass opacity, and then reticular opacity, or if you're in the idiopathic pulmonary fibrosis area, those RO areas would be called fibroblastic foci. And so the biggest difference in terms of what we'd want to be representing is that ground glass areas are more inflammatory, although have fibrosis as well, and potentially would have edema, tissue edema. And then reticular opacity, those are also sites of active remodeling, but are more fibrotic. So there's a bit of difference in terms of what we think of, and those are very broad brushstrokes, but what we need to represent in terms of translating the pathophysiology to the mathematical and computational representation. Those two have ongoing disease progression. And then in these patients for SSc-ILD, there's not as much honeycombing as there might be in IPF, but we do represent areas where there's collapsed lung function due to too much extracellular matrix buildup, and those have no contribution to respiratory function. And they're relatively rare in these patients. So those are the main areas that we need to consider in terms of representing what's going on in these patients. So how do we build that in terms of representing conceptually, but also computationally what we need to build in? Well, normal parenchyma, we have the alveolar epithelial cells represented. And then I'll go through ground glass opacity first, since the sort of structure is replicated. But we have inflammation here, neutrophils, macrophages, fibroblasts, so activation of resident to activated fibroblasts, and then ultimately collagen, elastin, and proteoglycan production and ECM deposition. And then we have ongoing alveolar epithelial cell injury, as well as endothelial cell contributions to disease pathophysiology. And that is some of the systemic connection associated with systemic sclerosis. So while we're modeling the lung, we do consider systemic effects. And then we represent kind of a mediator box. If we drew all those out, this would look like a big pile of arrow spaghetti. But all of these cell types are dynamically producing mediators that regulate the ongoing disease processes. So in terms of the mechanistic modeling, we're trying to capture all these interactions between cell types that ultimately allow us to model the ongoing pathophysiologic processes that will lead to a representation of clinical outputs. So I've built out this slide, but what I was trying to demonstrate here is that we also represent the reticular opacity. And while it looks the same, within the modeling, we're able to represent a more inflammatory zone in the ground glass opacity and a more fibrotic zone in the reticular opacity. And then we represent from reticular opacity to a honeycombed area. And then all three of those diseased zones contribute to clinical outputs, FVC and DLCO. And forced vital capacity, FVC, is one of the key clinical outputs that's often considered in clinical trials. So we would track that both from a model representation standpoint, but then also examining the difference between an untreated placebo group of patients and a treated group of patients. And one key aspect of the pathophysiology as well, before we move on, is that the ground glass opacity, in part because it's more inflammation driven, there's data to look at in terms of reversal, both from a ground glass opacity and reticular opacity standpoint. We did sort of look at whether we'd want to represent reversal from both zones, but certainly the more established, stronger representation would say that ground glass reversal is possible. And so that's what's represented in the model, although we're capable of representing both directions. And so, those are all the kind of design decisions that we include and consider when developing the model. And what ultimately drive our treatment responses. So that's the model itself. And then all of those interactions are established through examining preclinical data, clinical data, and we have an entire document, so you can imagine, that would establish the basis for including all of these different pieces. But ultimately, we needed to establish that all of those together compare well against available data and on a population level. So what we'll go through next are the simulated patients compared to those different clinical data sets. So here we're first looking at inflammation and fibrosis comparators. So in the first column, we have macrophages and neutrophils. And you'll note that there is a reasonable spread in each. In some cases, we're going beyond the clinical data. Some cases, we're getting a reasonable spread within the clinical data, and in both macrophages and neutrophils, we have a decent spread, which you want different individuals to be represented. And then in the right column, we have a collagen to elastin ratio, so that's a fibrotic output. And again, allowing some patients to represent the clinical data, and then have some expanding beyond, with the idea being that the model, we want to be able to represent what has been seen, but then also be able to examine scenarios mechanistically beyond what has specifically been observed. So the capability of these patients includes both of those aspects. And then we have endothelial cells as well, demonstrating some variability there within the clinical support, clinical data support. And then I mentioned we had one mediator box. These are critical to establish and regulate inflammatory processes. So say TNF-alpha being pro-inflammatory, fibrotic processes, TGF-beta, and then PDGF, MMPs, TIMPs also regulate aspects of fibrosis, and IL-6 regulates aspects of inflammation. And so there are simultaneous comparisons across all these different mediator axes against clinical data. So, we're not picking and choosing different comparisons. We're saying our patients across all of these are comparing well against the different data sets here. And so, and we also have comparisons as well against additional mediators. And some of these are getting more into the biomarker cases, so collagen biomarkers and inflammatory biomarkers like CRP. So ultimately, what we're trying to establish here is that the base model with available data compares well. And so our different patients are sitting at levels that are reasonable, and all of the components are interacting together well to establish patient variability, which then we ultimately should respond reasonably well under treatment. So those are the kind of deeper into the mechanistic underpinnings of the model. If we go that level up and think about the clinical outputs, our FVC, DLCO outputs, that's the aggregation of all of the underlying pathophysiologic components and manifested in terms of the patient disease state. So how are the patients, in this case, a percent predicted FVC? So given a patient's age, height, what would we predict their FVC to be? And then where are they actually sitting? And you can sort of, there are a large number of orange circles here, but you can sort of see that the cloud of orange circles for FVC is comparing reasonably well against the data cloud that's underneath it. And we're doing that both in terms of the available data under the upper left figure here for FVC and DLCO. So the data cloud's a little bit tighter there, but we're comparing well. And then there are some additional data sets as well that we're matching both the kind of average trend, but then also the overall data spread. You're never going to match the data spread exactly, but we've got a decent kind of overall population. So this is ultimately establishing that we have our patient population capturing the reasonable spread of the pathophysiology. And we took that base representation, and I think Lisa alluded to this earlier, that you also need to test your model against treatments. And so, here I've mentioned IPF and ILD and highlighted that in ILD, we've run the model against nintedanib, mycophenolate, and tocilizumab. And those first two, we also were using to help calibrate the model and make sure that our responses were correct. And then to some extent, a validation. So we were using the calibrated model and then validating the tocilizumab response. And so almost building that out like we would a novel treatment. And so, here we're just going to show the results of doing that. But the first nintedanib here, we simulate a cohort of 444. Now, how do we come up with that number? Well, that is matching clinical characteristics of the trial that we're comparing against. So we would match, say, available FVC, DLCO, age characteristics, if they're available, of the population, but then also match the overall trend of the placebo group. So that's why here these solid lines, in terms of the clinical data in the black solid line, and the simulation untreated group in the red solid line, match their trend. So we're selecting our simulated individuals to match the clinical trial untreated group. And then we apply treatment. Those are the dashed lines, the red dashed line. And we want to see, in general, that by, overall, but particularly by 52 weeks, that we're seeing a similar trend in terms of the difference in FVC from untreated to treated. So nintedanib, we're doing reasonably well, and there are some kind of differences due to co-administration of other drugs that can explain why we're not matching exactly. In terms of mycophenolate, the overall trend, so again, we're matching the patient groups, the trend of percent predicted FVC over that 52 weeks. And if you imagine these lines sort of shifted, they basically lie on top of each other. So we're predicting the treatment trends almost exactly. In terms of our SIM cohorts, in this case, 71 individuals versus the treatment group of around, again, 70 individuals or 80, depending on the group. So in terms of our treatments, these are more helping us calibrate the model. We're doing a reasonable job predicting the treatment response for mycophenolate and nintedanib. And then tocilizumab. So those first two, I think, were the ones in the actual poster. But we also wanted to show tocilizumab here as a demonstration of a bit more of a validation response, in the sense that we were using the first two to help iterate on the model development. And tocilizumab was more, let's match the placebo group and parameterize the tocilizumab mechanism of action, which is IL-6 inhibition, and see how well we're doing against clinical data, and you can sort of see the response is fairly good here. So that gives us more confidence that modeling a novel treatment, modeling a novel treatment, that we're capturing the pathophysiology reasonably well. So if we need to implement a new mechanism of action, that we would be able to leverage the existing pathophysiology forward. And so, we've done some of that with proprietary compounds in ILDsym. We've also done that in IPFsym, which is a very similar platform. So there's reasonable confidence in both platforms that we'd be able to model a novel treatment successfully. So that was, kind of a whirlwind overview of ILDsym. But the work from the development team, who are listed here, was phenomenal in bringing this platform to where we have it. And certainly we look forward to evaluating novel targets with this platform. And then this is also well poised to examine other interstitial lung diseases beyond systemic sclerosis associated ILD. So I think that is sort of ultimately the poster, and then looking forward.
Thank you so much, Zach. We have a minute left. I'm going to try to do a single Q&A question. Not sure if we're going to have time. If we don't have time and we have to cut it early, we can save it for the Q&A section later. The question is, how strong is the correlation between FVC and disease extent? What kind of regression, i.e. linear, exponential, etc., would fit in?
So ultimately, it was fit using an exponential, and it's a reasonably strong correlation. So I'm going to try to sneak back real quick. So that red line is the correlation line.